Good morning, good afternoon, and good evening, everyone, and welcome to our global webinar series hosted by Eqra Science Bioinnovation around the world. Joining us today for this advanced session on organelle crosstalk, mitochondria, and lysosomes in immune cell fate and function. My name is Dr. Sundas Qureshi, and I will be your host for today's webinar. Over the next hour, we will explore the critical interactions between mitochondria and lysosomes and how they influence immune cell behavior, metabolism, and function. Before we dive into today's topic, I'd like to briefly introduce Eqra Science. Founded in 2009, Eqra Science is now a global life science company with a presence in over 150 countries. Multiple international certifications have been obtained, including ISO 9001, ISO 13485, and CE certification, and tens and thousands of product citations in internationally recognized peer-reviewed journals, including Nature Group, Cell, and Science. Since launching our first bioresearch reagents and ELISA kits in 2011, we have continuously expanded our portfolio. Cell culture solutions and clear kits in 2013, antibodies, proteins, immunohistochemistry and Western blot kits, metabolism assay, food safety, and animal disease kits, and flow cytometry antibodies over the following years. Most recently, we introduced advanced tools such as induced differentiation media, exosome isolation kits, ELISpot, CellaQuant, and primary cell isolation kits, while also earning recognitions like CITAB Supplier to Watch and Kotler Award for Customer Value. This timeline reflects our ongoing commitment to innovation and high-quality tools supporting cutting-edge research, including studies on redox biology, which we will focus on today. Okay, before we begin, a few housekeeping notes. Please keep your microphone muted during the presentation and submit any questions via Q&A panel. We will address them at the end, and for other issues, please write in the chat box. A digital participation certificate and a session recording will be sent to all registrants. Attendees also have the chance to receive free samples of selected Eqra Science products. Details are on this slide, and the list with eligibility and claim instructions will be sent to your registration email. Now, it's my pleasure to introduce today's speaker, Dr. Eqra Hafeez, who will take us through the fascinating mechanism of organelle crosstalk in immune regulation. Dr. Hafeez is a biologist specializing in cellular and molecular mechanism of aging, stress resistance, and metabolism. Her PhD work in Drosophila melanogaster used advanced transcriptomes and metabolomics to study intracellular signaling and organelle function. With a strong background in immunohistochemistry, enzyme assays, and molecular biology, she brings deep expertise in a crosstalk between mitochondria and lysosomes, adding a broad biological perspective to today's discussion on immune cell fate and function. Dr. Hafeez, over to you.
Thank you, Dr. Sundas, for giving me this opportunity. Thank you, Dr. Sundas, for giving me this opportunity to talk about organelle crosstalk with special focus on mitochondria and lysosome in immune cell fate and function. Good morning, everyone. First, I would like to say thank you all for joining today's session. In this webinar, we will explore how communication between two key organelles, mitochondria and lysosome, shapes immune cell fate and function, and how this intracellular communication offers new insight for therapeutic strategies. If you have any technical question regarding this webinar, you can contact me at the provided email address. Let us begin this session by outlining a complete roadmap focused on organelle crosstalk with particular emphasis on mitochondrial lysosome communication. In this session, we will move step by step from fundamental mechanisms to disease relevance and finally therapeutic way forward. Firstly, we will discuss how and why mitochondria and lysosome communicate and explore the molecular and cellular mechanisms that enable this interaction. Mitochondria and lysosome communicate through specialized contact sites that integrate metabolic and degradative function. Mitochondria supply energy and metabolic signals, while lysosome control recycling and signaling through hydrolytic activity. At mitochondrial lysosome contact sites, the exchange of metabolites, lipid transfer, and calcium synchronize organelle function and maintain cellular homeostasis. In short, this dynamic crosstalk form a signaling hub that enables cells to adapt metabolism and withstand appropriate stress responses. Signals driving mitochondrial lysosome communication helps organelle to crosstalk functional maintenance and metabolic balance. In the case of damaged mitochondria, stress signals such as ROS are generated, which act as alerts to lysosome, initiate mitophagy, and help maintain redox homeostasis. In parallel, lysosome release calcium ion signals at contact sites that regulate mitochondrial metabolism, ATP production, and fission-fusion dynamics. If we talk about ATP level, they reflect mitochondrial energy status and are sensed by lysosomes to modulate mTOR-C1 signaling, trigger autophagy, and enable the efficient turnover of dysfunctional mitochondria. Mitochondrial lysosome crosstalk matters because this communication is essential for maintaining cellular homeostasis. In this crosstalk, lysosome ensure proper mitochondrial function by regulating mitochondrial turnover and recycling. And in return, mitochondria supply the energy required for lysosomal degradation process. In addition, mitochondrial lysosome communication also play a critical role in immune and stress responses such as T lymphocyte, neuron, and other highly specialized cell types rely on this interaction to rapidly adjust their metabolism, withstand stress, and respond effectively to activation, infection, or injury. Beyond basic maintenance, this crosstalk is crucial for cellular quality control. In this situation, damaged or dysfunctional mitochondria are recognized and selectively delivered to lysosome for degradation, which prevent the accumulation of harmful byproducts that could impair cellular function. When immune cell is under stress, it increases oxidative stress and mitochondrial reactive oxygen species production through pathways such as protein kinase C beta P66 and also enhance the calcium ion influx. This excess ROS and calcium ion triggers mitochondrial dysfunction, including opening of the mitochondrial permeability transition pore. Mitochondrial permeability transition pore and loss of membrane potential, creating a critical decision point between cell death and survival. Then the damaged mitochondria are selectively recognized by the PINK1 and PARKAN pathway, which labels them for degradation by mitophagy, and these mitochondria are engulfed by autophagosomes that subsequently fused with lysosomes where mitochondrial components are degraded and recycled. So this coordinated mitochondrial lysosome interaction limits excessive inflammation, prevents release of mitochondrial danger signals, and regulate immune cell survival and functional adaptation during the stress. As we observe the functional interdependence between mitochondria and lysosome drive disease development and progression, therefore let's focus on how these crosstalk contribute to pathology and explore potential therapeutic strategies to restore organelle function and overall cellular health. Mitochondrial lysosome contact sites are disrupted in many neurological and lysosomal disease, connecting specific genes or proteins to changes in these contact sites and their role in disease development. Like for example, Parkinson's disease is associated with mutations or dysfunction in genes such as GBA1 and alpha-synuclein etc. These changes lead to prolonged or reduced mitochondrial lysosomal contact site and altered calcium ion handling, and in some cases fewer contact site overall. Another example is CMT disease, which is linked to genes like MFN2 and RAB7, etc. Shows impaired regulation, disrupted lysosomal dependent mitochondrial messenger RNA translation, and changes in the number of contact site and redox dependent defects. On the other hand, lysosome storage disorder involve TRPML1 and NPC1 channel. This disorder exhibit altered mitochondrial lysosomal contact, calcium signaling, and often an increase in the number of contact site. Finally, epilepsy is associated with TBC1D15, which is characterized by excessive calcium ion accumulation at mitochondrial lysosomal contact site. Mitochondria and lysosome normally communicate through mitophagy, mitochondrial-derived compartment, mitochondrial-derived vesicles, and membrane contact site to maintain cellular health. But in case of stressed mitochondria, or when mitochondria become damaged and their membrane potential drop, pink one becomes stable on their surface and activate enzymes such as PKN and GBA that tag the mitochondria for degradation by lysosome. During this process, MDCS and MDVS are released to signals lysosome to increase their activity. But if few lysosomes are faulty and unable to fully process the mitochondrial cargo, this failure leads to the accumulation of alpha-synuclein, which clumps together to form Lewy bodies, a key feature of Parkinson's disease, ultimately contributing to neuronal dysfunction and degeneration. MFN2 and GDAP1 are two proteins which are essential for healthy mitochondria in peripheral neurons at mitochondrial lysosomal contact site. Any mutation in this protein at mitochondrial lysosomal contact site disrupt the whole process and leading to fragmented, poorly distributed mitochondria and impaired communication with lysosome. As a result, ATP production is reduced and ROS increases, causing neurodegeneration and drive the characteristic symptoms of this CMT disease, like progressive muscle weakness, sensory loss, gait abnormalities, and limb deformities. Normally, when the cells need metabolic support, lysosomes store calcium ion and release it through TRPML1 channel. This calcium ion is directly transferred to nearby mitochondria at mitochondrial lysosomal contact site and enter mitochondria through VDAC1 and through MCU complex channel. After calcium ion uptake, mitochondria activate key metabolic enzymes and maintain mitochondrial function through ATP production. In case of ML4 mucolipidosis type 4 mutation in TRPML1 disrupt lysosomal calcium ion release and prevent efficient calcium transfer to mitochondria. As a result, mitochondria metabolism declines, which contribute to neurodegeneration, muscle weakness, vision impairment, and distinct facial features in affected children. Under normal condition, lysosome release calcium ion through the TRPML1 channel, and this calcium is taken up by mitochondria to support energy production and regulated mitochondrial dynamic. In contrast, mutation in TBC1D15 are conditions such as seizure and chronic neuronal stress disrupt this communication. Excessive lysosomal calcium ion transfer then lead to the mitochondrial calcium overload, resulting impaired oxidative phosphorylation, elevated oxidative stress, and mitochondrial dysfunction. This mitochondrial failure promotes neuronal injury and established a feed-forward loop that facilitates focal seizure activity and progression to generalized epilepsy. Mitochondrial lysosome contact site have an important role in regulating glucose uptake by controlling the trafficking of GLUT4 vesicle. RAB7, a key regulator of late endosomal and lysosomal pathway, direct GLUT4 vesicles through endosome maturation, sorting, or endocytic trafficking, which ensure they reach the plasma membrane. TBC1D15, a GTPase activating protein for RAB7, which fine-tune this process by regulating RAB7 activity and making sure GLUT4 vesicle follow the correct route. This coordination at the mitochondrial lysosomal contact site link energy status At mitochondrial lysosome contact site link energy status with vesicle trafficking and allow cell to efficiently move glucose into the cytoplasm and maintain metabolic balance. When mitochondrial lysosome contact sites are not functioning properly, the trafficking of GLUT4 vesicles to the plasma membrane is disrupted. This means that glucose cannot enter muscles and fat cells efficiently, even when insulin is present. As a result, blood glucose level remain high and the pancreas compensate by producing more and more insulin. Over time, this leads to insulin resistance and contributing to metabolic disease with symptoms such as increased craving for food, carbohydrate inflammation, and chronic carb consumption. mTOR-C1 on lysosome promotes cholesterol biosynthesis through SCK1 and control cholesterol trafficking to mitochondria through NPC1, TSPO, and star D3, a cholesterol transporter at mitochondrial lysosomal contact site. When this pathway is overactivated, cholesterol starts to build up in mitochondria and lysosomes, impairing fatty acid breakdown, generating oxidative stress, and disrupting mitochondrial function. As a result, in the liver, these changes prevent proper fat clearance, leading to lipid accumulation in hepatocyte and contributing to the development of non-alcoholic fatty liver disease. Now we will go through key case studies highlighting mitochondrial lysosome crosstalk in T cell and in cancer. We will focus on when this interaction is activated and how it influences cellular metabolism, survival, and functional outcome in this experimental condition. Mitochondrial lysosome interactions are most prominent during T cell development, activation, and differentiation, as well as during metabolic stress, the formation of regulatory T cells and memory T cells, when energy demand and cellular remodelings are highest. Furthermore, dysregulation of this interaction contribute to disease and dysfunction, cellular dysfunction, including chronic infection, autoimmunity, and cancer. Mitochondria and lysosome communicate through bidirectional metabolic signaling in T cell. Lysosome function as nutrient sensing platform that detect amino acid and glucose, regulating the AMPK pathways to promote catabolism and activating mTOR-C1 signaling to drive anabolic process. They also influence gene expression through TFEB, which induce lysosomal biogenesis and links metabolic status to cellular clearance and energy homeostasis. Mitochondria generate ATP and key metabolic intermediate through the TCA cycle and oxidative phosphorylation, while also producing mitochondrial reactive oxygen species and metabolites such as citrate and S-adenosyl methionine that modulate nuclear acetylation and methylation. Together, these mitochondrial signals couple cellular energy and redox status to NFAT-dependent transcription and epigenetic remodeling, illustrating how mitochondrial activity shapes T-cell immune responses. During CD4 positive T cell activation, mitochondrial respiratory complex 1 to 5 are tightly regulated to support ATP production and biosynthesis. And signal from mitochondria such as ATP level communicate metabolic status to lysosome, which in turn regulate nutrient recycling and ensure removal of damaged mitochondria, while sustaining energy demand required for CD4 positive T cell survival, activation, and proliferation. In case of CD8 positive T cell, mitochondrial fusion dynamics determine metabolic fate, with fusion promoting oxidative phosphorylation and memory differentiation. And in case of fusion derived glycolysis and effector cytotoxicity, lysosome mediates sensing of mitochondrial stress trigger mitophagy, enabling mitochondrial lysosome crosstalk to preserve metabolic fitness and regulate CD8 positive T cell function. In case of regulatory T cells, Tregs depend on mitochondrial oxidative metabolism, with the TCA cycle and electron transport chain complex 1 to 4 during oxidative phosphorylation and ATP production. Under the control of LKB1, mTORC1, TFAM, and MYC. This metabolic program upregulate the expression of ICOS, CTLA4, and KLRG1, which promotes interleukin 10 secretion and establish mitochondrial oxidative phosphorylation as a central hub for Treg functional competence and immune tolerance. Effective T cell activation and differentiation depend on the integration of four distinct signals. First one is antigen recognition, where T cell receptor recognize cognate antigen which initiate T cell activation. Co-stimulation, which validate antigen recognition and drive full activation by promoting metabolic engagement. Third one is cytokines, where cytokines cues instruct T cell differentiation by shaping transcriptional and metabolic programs. Last one is nutrient uptake, where nutrient availability supply the energy and substrate required to execute activation and differentiation program. As a result of these four signal, mitochondrial lysosome interactions are reprogrammed in process called metabolic rewiring. This coordination and metabolic signaling supports T cell functional adaptation and enables effective immune responses. Mitochondria and lysosome signaling play a key role in maintaining Treg cell metabolism and function. For example, when the mitochondrial protein OPA1 is removed from Treg cells, their mitochondria become stressed. This stress causes the cells to make too much reactive oxygen species and ATP, which disrupt their normal metabolism. As a result, Treg cells lost their ability to suppress immune responses, and this leads to excessive inflammation. This happens because mitochondrial stress activates major metabolic signaling pathway in the cells, including mTORC1, AMPK, and the transcriptional factor TFEB, which changes how the cell function. Similarly, when the lysosomal protein FLCN is deleted, Treg cells show similar problem to those seen with OPA1 loss. These cells enter a kind of abnormal low activity metabolic state, meaning they cannot properly survive in tissue or control immune responses. Because of this, Treg fail to accumulate where they are needed and cannot suppress inflammation. Interestingly, when TFEB is also removed, these defects are corrected, showing their communication between mitochondria and lysosome is crucial. Overall, this means that proper coordination between mitochondria and lysosome is essential for Treg cell development, function, and for maintaining immune system balance. Imagine a strategies that take down cancer cells by cutting off their internal communication lines. That's exactly what happened here. Fullerenol, the allotropes of carbon, target lysosome, damaging their membranes and creating stress signals that ripples through endoplasmic reticulums and mitochondria. Normally, mTOR inhibitors activate and amplify autophagy. Then impaired lysosome prevent their function, prevent their fusion with autophagosomes. Thus, autophagy is paralyzed along with autolysosome accumulation. As a consequence, cancer cells are unable to restore subcellular homeostasis. Thus, the loss of communication between these key organelles, lysosome, endoplasmic reticulums, and mitochondria, pushes the cell into panoptotic death, a powerful combination of multiple cell death pathways such as pyroptosis, apoptosis, and necrosis. So, the selective disruption of lysosome, endoplasmic reticulum, and mitochondria. Crosstalk can effectively kill cancer cells and produces strong anti-tumor effects. Summarizingly, targeting metabolic pathways linked to mitochondria, autophagy, and lysosome can strategically reprogram T cell function in immune mediated disease. Modulating receptors such as KLRG1, ICOs, and CTLA4s alters mitochondrial metabolism to enhance T cell activation, proliferation, migration, and effector cytokine production, thereby strengthening anti-tumor immunity. Similarly, inhibiting autophagy related genes decrease autophagy flux in intratumoral CD8 positive T cells, which reduces immunosuppressive Treg accumulation and boosting anti-tumor responses. Further enhancing lysosomal biogenesis through FLCN supports the differentiation and migratory capacity of CD8 positive tissue resident memory T cells and helpful in improving and control of intestinal infections. When we look at immune cells like T cells, their fate and function really depend on the conversation between mitochondria and lysosomes. By checking mitochondrial health, autophagy and mitophagy activities, inflammatory signals, and modulating mitochondrial lysosome contact site, we can see how these crosstalk guide their responses using reliable kits like ELAB science. We can track these pathways and get reproducible insight into how metabolism shapes immunity. Thank you all for joining us. I hope this session provided a clear overview of mitochondrial lysosome crosstalk and from fundamental mechanisms to disease relevance and therapeutic perspective.
All right. I think you haven't heard me before. First of all, I would like to thank Dr. Eqra Hafeez for that insightful presentation on mitochondria lysosome crosstalk and its impact on the immune health. I hope I'm audible to all of you right now. If anyone can hear me, please write in the chat box. All right. Following the discussion by Dr. Hafeez, I'd like to introduce some of the ELAB Science products designed to explore these organelle interactions in the greater detail. On the left here, we have mitochondrial functional assays, including kits for the membrane potential, permeability transition pore, OCR measurement, and some live cell probes. These are ideal for monitoring the mitochondrial health and dynamics. On the right-hand side here, we have energy and reactive species assays covering ATP, ADP quantification, ROS detection, and calcium signaling. Together, these assays provide a comprehensive toolkit to study the cellular metabolism, oxidative stress, and organelle function. Building on that, we also offer a wide range of biomarkers and some flow cytometry tools, which are available as antibodies and are relevant to the organelle crosstalk and immune research. Cytokines and immune biomarkers include IL-10, IOCS, and some other biomarkers, which are available as antibodies or ELISA kits to measure the cytokine levels and receptor activity in immune cells. Mitochondrial and lysosomal biomarkers include MMP1, MMP9, MMP13, LC3, ATG7, and caspase activity kits, which are usable and useful for tracking the autophagy, organelle stress, and cell death. Over 4,500 flow cytometry options include both the flow cytometric antibodies as well as purified antibodies for the flexible labeling and analysis. As a reminder, all the webinar attendees are eligible to request one free sample from these products. Full details about the claim process will be sent to your registration email. Now we will move on to the question and answer segment. Please type your question in Q&A panel and we will discuss as many as possible. Okay, Dr. Eqra, we have a first question here. Can same JC1 kit be used for mouse CD8 positive T cells and human PMBCs? Can you please answer that? Unmute your mic first.
Okay, I will answer you. Yes, the same GC1 kit can be used for both mouse CD8 positive T cell and human peripheral blood mononuclear cells. Because human peripheral blood mononuclear cells, T cells, you need to use fewer human cells per well to get a clear signals. Otherwise, the staining procedure and analysis are the same for both cell types.
Okay, if you have any questions, please write in the Q&A box. Dr. Eqra, please unmute your mic.
Okay.
All right, so the next question we have here is what molecular switch turns on AMPK off during the chronic stress?
Okay, the answer of your question is that,
please hold on.
Okay, the answer of your question is that during chronic stress, calcium ion release from lysosome activate calcineurin. Calcineurin then turn off AMPK by removing a phosphate from it at THR 172.
Okay, let me see if we have any more questions here. Okay, Miss Neha just have asked the answer. Asked one question. I have tried the OCR kit. However, as per the kit manual, I'm unable to set the range for linear par, like where to start and end. Miss Neha, I would like to answer this question for you. Actually, for this kind of question, because it needs a detailed explanation, so would it be possible for you to write to us in the email so that we will help you with the detailed answer? Secondly, I have noted down your question and we will give the detailed reply to you in the email. Okay, we have one more question from Alvin Alexander in the chat box. And this is about PPT might not be distributed, but we will share the recording of this webinar to you. Okay, so Dr. Eqra, next question we have here is, will the systemic ATG5 inhibition cause the autoimmunity? Actually, this is a kind of gene question. So, Dr. Eqra, can you please look into it? Will the systemic ATG5 inhibition cause Okay, so let me repeat the question. It is, will the systemic ATG5 inhibition cause the autoimmunity?
So the answer is that blocking ATG5, a gene important for autophagy, usually does not cause autoimmunity. Short-term or T-cell specific inhibition has been shown to be safe and the immune system continues to function normally without attacking the body. However, completely removing autophagy for a long time in certain immune cell could potentially affect immune balance. So short-term or targeted ATG5 inhibition is generally safe and does not trigger autoimmune reactions.
There is another question. In the cancer PDAC, there is a crosswalk between macroprensome and lysosome. Will some of your kid be helpful in studying their crosstalk? Okay, let me answer this question for you. Actually, this question again is detailing about the kids. So we will mark this question and we will answer you in the email. Let us check in our list. And if there is any such kind of kit available, we will definitely get back to you through email. And if possible, please write to us on events at elabscience.com. Okay, Dr. Eqra, we have another question here. How can I directly test whether mitochondria lysosome contacts are disrupted? Can you please answer that?
Okay.
Unmute your mic first.
You can perform live cell confocal or super resolution imaging with fluorescent marker for mitochondria and lysosome. And you can quantify contact frequency, duration, or proximity using time-lapse analysis or dedicated contact site quantification software.
Okay, let us see if there is another question. Okay, here we have another question. Which assay is most suitable to study mitochondrial lysosome crosstalk in immune cells? Okay, Dr. Eqra, are you going to answer this or should I answer this question?
Okay, I answer this.
Okay, all right. Unmute your mic first.
So the answer of your question is that a combined approach works best. Mitochondrial membrane potential GC1 OCR assay and MPTP kit assesses mitochondrial function, while LC3 ATG7 and caspase activity markers help monitor autophagy, lysosomal activity, and cell death. Together, these assays provide a functional view of organelles interaction.
Okay, let me see if we have any more question here. Yeah, there is another question. What signaling molecules coordinate mitochondria lysosome crosstalk? Okay, let me answer this question. Basically, the primary signaling molecules coordinating the RAB7 GTPase, which Dr. Eqra also mentioned in her presentation. Then there is phosphatidylic acid, which is PA, and calcium ions, some of the reactive oxygen species, and mTORC1 and AMPK. Okay, Dr. Eqra, there is another question we have. Okay. Flow cytometry. This question is about flow cytometry.
Okay.
Question number 3. Okay. How can flow cytometry help in studying immune cell fate related to organelle function?
Okay. The answer is that flow cytometry enables multi-parametric analysis of immune markers such as interleukin 10, CTLA4, ICOS, and KLRG1 at the signal cell level. When combined with functional assay, it allows correlation between metabolic state activation and immune cell differentiation.
Thank you so much. Now we have another question. Okay, you just mentioned few products. No, I mentioned, and I think they are referring to those products. Are these products suitable for studying immunometabolism and metabolic reprogramming? Actually, I can answer this question. Yes, these assays will be used to measure OCR, ADP ratio, ROS, and calcium flux, and the mitochondrial stress to particularly useful for the immunometabolism studies. Ultimately help us to link the metabolic changes with immune activation and exhaustion. Okay, we have one more question. Okay, it's here. Can these assays be used in primary immune cells such as T cells or macrophages? Thank you.
Yes, many of the mitochondrial ROS, ATP, and flow cytometry based assays are suitable for primary immune cell, including T cell and macrophages, provided that cell number and assay conditions are optimized.
Okay, so the main issue here we have is that the assay conditions must be optimized while you are dealing with primary immune cells such as T cells or macrophages. Okay, one question is that where we will get the e-certificate. Okay, actually, I have mentioned in the beginning that e-certificate will be sent to your email IDs, which you have used to register for this webinar. So stay tuned. We will send the recording as well as the participation certificate and the free claim form, free sample claim form for you on your registration email ID within 2 weeks. The question is, can I request technical guidance when selecting the right products? Definitely, absolutely. eLab Science provides the technical support to help select appropriate assays or antibodies based on your own application. So you can contact our technical support team via techsupport@elabscience.com guidance. But if you don't want to connect with your local distributor regarding your technical queries, you can directly write to us at techsupport@elabscience.com, or you can also contact us at events@elabscience.com. Here it is mentioned on this slide. All right, guys, we time is short, so let's wrap up. Okay, thank you all for joining us and engaging with us in this session. We hope Dr. Hafeez's presentation, along with the overview of eLab Science relevant products, provided the valuable insights into organelle crosstalk and immune cell regulation. If we are unable to address any of your questions during the session, please feel free to send us those questions on events@elabscience.com or techsupport@elabscience.com. Our team will provide you with detailed responses. And don't forget to check your registration email for the details on claiming your free sample and accessing the session recording. We will also send the participation certificate to all the attendees on their registration email. We look forward to seeing you in our future webinars. Have a great day and happy experimenting.